Good day, dear student. Lab 5 will be about Streptococcus species. I will explain each slide in a simple way. We will connect the ideas together so we can understand not only the information but also the reason behind each laboratory test and how we make the diagnosis and how can differentiate between the species in the. Now, Streptococci are among the most important bacteria in medical microbiology. It's responsible for many infections from the mild sore throat to the life-threatening disease like pneumonia, sepsis, and meningitis. In this lab, we will focus on a general characteristic, classification, and the most important point, how to identify them in the lab. I will start with the general characteristic. It's a gram-positive cocci. It means it appears under the microscope in a blue or purple color, and the shape is cocci and arranged in pairs or chain. The arrangement is chain or maybe diplococci. The catalyst is negative, and this is very important because we can differentiate between streptococci, that is catalyst negative, from staphylococci, catalyst positive. So streptococci is catalyst negative. It means they don't have the catalyst enzyme. Non-spore forming bacteria and non-motile and produce the capsule, which increase the virulence. Many strains have a capsule, such as Streptococcus pneumoniae capsule that help to avoid the phagocytosis. Facultative anaerobic, it means it can grow in both presence or absence of oxygen, and it's sensitive to dry and heat. It means they don't survive long outside the body, and many are fastidious. It means they require enriched media in the lab, like blood agar. Now, after understanding the general characteristic, as I mentioned, it's a gram-positive cocci and arranged in a chain under the microscope. Now, the culture characteristic is very important, and we can classify the streptococci according to the type of the hemolysis and blood agar, as streptococci grow in blood agar and produce different type of the hemolysis. Some produce the alpha hemolysis, some strains such as Streptococcus pneumoniae and viridin streptococci, and the growth it appears as a green zone around the colony due to the partial destruction of the red blood cell. Others produce the beta hemolysis such as Streptococcus pyogenes and Streptococcus agalacti, and the growth appears as a clear zone around the colony due to the complete destruction of the red blood cell or complete hemolysis. Another group produce the gamma hemolysis such as Enterococcus species, Enterococcus faecalis and Enterococcus faecia, and that means they are non-hemolysis and they are no destruction of the red blood cell on blood agar. In this image, you can see the three types of the hemolysis of the streptococci on blood agar. The first image, they are no change on blood agar. They are non-hemolysis, which is type of the gamma hemolysis. And in the second image, they are clear zone around the colony. And this type, it's beta hemolysis due to the complete destruction of the red blood cell. And the third image, they are green zone around the colony due to the partial destruction of the red blood cell. And this type is alpha hemolysis. Here they are another image of the type of the hemolysis of the streptococci on blood agar. The beta hemolysis that indicate by the clear zone around the colony due to the complete destruction of the red blood cell. And alpha, they are indicated by the green zone around the colony due to the partial destruction of the red blood cell. And the gamma hemolysis, they are non-hemolysis of the red blood cell on blood agar. Today in this video, I'll go to show you the three types of the hemolysis on blood agar and the difference between them. Okay, the first one here, this is alpha hemolytic Streptococcus pneumoniae. You can see here they are green zone around the colony, that is alpha hemolytic, which is due to the partial destruction of the red blood cell on blood agar. This is after streaking method and incubated the plate with increased CO2. You can see after 24-hour incubation, you can see the green zone around the colony, which indicates the alpha hemolysis on blood agar. The next plate is beta hemolytic group B. Remember, group A and B, both of them produce beta hemolysis on blood agar. And you can see here they are clear zone around the colony. But I will go to show you the difference between the group A and group B in blood agar how to produce the beta hemolysis. Here the clear zone around the colony is usually small. But in group A, beta hemolytic, which includes Streptococcus pyogenes, you can see how the clear zone around the colony is usually large, which is indicate the complete destruction of the red blood cell on blood agar. The third type is gamma hemolysis, which include enterococci. You can see they are growth of the bacteria on a plate after incubation 24 hours with increased CO2, but they are no zone around the colony. You can see the colony, they are no zone around the colony, which is indicate the gamma hemolysis. This is all about the three types of the hemolysis. Now, another classification of the streptococci is according to the presence or absence of C-carbohydrate antigen on a cell wall. Rubica Lancefield classifies streptococci in a two group according to the presence or absence of this antigen on a cell wall. That's why we call the Lancefield grouping. And the two group is groupable streptococci, that means they have the C-carbohydrate antigen on a cell wall. And the most important group is group A, Streptococcus pyogenes, and group D, Streptococcus agalactiae, and group T, Enterococcus. And the non-groupable streptococci, that means they don't have the C-carbohydrate antigen on a cell wall, such as Streptococcus pneumoniae and viridin streptococci, like the Streptococcus mutans that cause the dental caries. Now, after classification according to the Lancefield grouping and according to the hemolysis on blood agar, we discuss each group. As I mentioned in a theory lecture, Streptococci is a large group of the bacteria, therefore single system is not enough for classifying them. And this classification is very important. It allows to identify the microorganism in a lab. Now, the group A, beta hemolytic Streptococci, which includes Streptococcus pyogenes, we can identify in a lab. If we take the throat swab, because it's the most common cause of the pharyngitis and tonsillitis, if we take the throat swab and we done the gram stain, it appeared like a gram-positive bacteria. It means it appears blue or purple under the microscope, and it's cocci, it means it's spherical under the microscope and arranged in a chain. When they grow on blood agar, it produces the beta hemolysis. That means they are clear zone around the colony, and the colony is usually small and transparent. After the microscopic examination and the culture characteristic, we do the biochemical test. And all species of the streptococci is catalyst negative. That means they don't have the catalyst enzyme. And when we done the bestracin sensitivity test, we can differentiate between the two beta hemolytic streptococci, Streptococcus pyogenes and agalactiae, as Streptococcus pyogenes is sensitive or susceptible to the bestracin antibiotic test, while the agalactiae is resistant to the bestracin antibiotic. Now, the group B beta hemolytic Streptococcus agalactiae is also beta hemolytic, but it's different from the group A beta hemolytic. It appears under the microscope, also it's gram-positive and cocci and arranged in a chain when we done the gram stain. Regarding the culture characteristic, when we grow on blood agar, it produces the beta hemolysis, but it's usually narrow beta hemolytic, and produce gray-white colony and mucoid appearance, because it's capsulated microorganism and the type of the capsule is polysaccharide capsule, unlike to the group A that also have the capsule, but the type of the capsule in a Streptococcus pyogenes is hyaluronic acid capsule, while here it's a polysaccharide capsule. That's why it usually have the mucoid appearance. When we done the biochemical test, as I mentioned, all species is catalyst negative, and when we done the bestracin sensitivity test, it's bestracin resistant. And in a COMP test, it's positive COMP test. That's mean they have the COMP factor. I already explained the bestracin sensitivity test and the COMP test in a next slide. And usually it causes the neonatal meningitis and sepsis. That's why the pregnant women are screened for group B streptococci. Bestracin sensitivity test is test used to differentiate between the Streptococcus pyogenes group A from the group B Streptococcus agalactiae, which is both of them produce the beta hemolysis on a blood agar. How can differentiate between them? By bestracin sensitivity test. And the principle of this test is the low concentration of the bestracin can inhibit the growth of the Streptococcus pyogene and give the zone of the inhibition around the disk of the bastracine, and the procedure it inoculated the blood agar by the spreading method on a plate and put the bastracine disc and incubated 24 hours at 37 Celsius. After that, we look at the zone of the inhibition. If zone is present, it indicated the group A Streptococcus, which is Streptococcus pyogenes. If there are no zone of the inhibition around the disc, it indicates Streptococcus agalactiae. Now, the CAMP test is specific for group B Streptococci, specific for Streptococcus agalactiae, as the group B produce the CAMP factor that enhance the hemolysis caused by Staphylococcus aureus. When striking perpendicular, arrow shape enhance hemolysis appearance, and this is the positive test. This confirms Streptococcus agalactiae. I already showed you the video about this test. The procedure of the CAMP test is very simple. We take the straight line of the Staph aureus on a blood-shaped agar and then take the test Streptococcus and straight it perpendicular at it and leave a gap between the two streets and incubate it at 37 for 24 hours. After that, we observe the result. The arrow shape, it indicates the positive result. And here in this image, you can see the positive result against the negative result. The positive result is indicated by the arrow shape, which is indicates Streptococcus agalactiae, and the negative it indicates the Streptococcus pyogenes. Why this happen in a positive result? The arrow shape it indicates the beta hemolysis that enhance in the presence of the Staph aureus by producing the CAMP protein by Streptococcus agalactiae. So the CAMP protein is produced by the Streptococcus agalactiae and enhance the beta hemolysis of the Staph aureus, and the positive result appear like the arrow shape, while the negative result, they are no CAMP factor and the hemolysis will not be enhanced, which is indicates Streptococcus pyogenes. So the principle is Streptococcus agalactiae produce the CAMP factor, extracellular protein that enhance the beta hemolysis of the Staph aureus and enhance the hemolysis on blood agar, while the pyogenes they don't have the CAMP factor. Now we will move to the alpha hemolytic streptococci that includes Streptococcus pneumoniae and viridin streptococci. Remember, beta hemolytic produced by pyogenes and agalactiae, while alpha hemolysis by Streptococcus pneumoniae and viridin streptococci, gamma hemolysis by the enterococci. Alpha hemolytic now by Streptococcus pneumoniae. Streptococcus, it appears under the microscope as gram-positive bacteria and arranged in a lancet-shaped diplococci under the microscope. As I mentioned, in a general characteristic, it may arrange in a chain or diplococci, because some species like Streptococcus pneumoniae, it arranged as a diplococci lancet-shape under the microscope. On blood agar, it produces the mucoid colony surrounded by green alpha hemolysis, and usually it forms smooth gelatin mucoid colony, which is reflect the partial hemolysis activity, and according to the biochemical test is catalyst negative, as all species of the Streptococci is catalyst negative. Regarding the optogene sensitivity test, is susceptible to the optogene antibiotic disc. This helps to differentiate it from other alpha hemolytic streptococci such as viridin group. And the mucoid appearance is due to the polysaccharide capsule, which is the virulence factor of this bacteria. And this helps all these characteristics is used in lab to diagnose of pneumococcal infection like pneumonia, meningitis, and otitis media. Now, the optogene sensitivity test used to differentiate between the two alpha hemolysis streptococci, Streptococcus pneumoniae, and viridins. And the principle of this test, the optogene can inhibit Streptococcus pneumoniae at the low concentration, and this test is essential in respiratory sample. When we inoculate the blood agar with the microorganism and apply the optogene test at the center of the plate and incubate it at 37 Celsius for 24 hours. After the incubation, we measure the zone of the inhibition around the disc. If the zone of the inhibition is equal or larger than 40 mm, it indicated the positive result. That means the organism is susceptible to this disc and indicate the positive result. While if the zone is less than or equal to 13 millimeter, it considered as a negative. It indicates the microorganism is resistant to the optogene. And the Streptococcus pneumoniae is sensitive, which is produced the zone equal or larger than 40 millimeter, while the viridans is resistant to the optogene antibiotic disc. Here we can summarize the practical identification strategy. In the first table, you can show the difference between the both beta-hemolytic streptococci, Streptococcus pyogenes, and Streptococcus agalactiae, as both of them produce the beta-hemolysis on blood agar, but the Streptococcus pyogenes is susceptible or sensitive to the bastracine antibiotic test, and it is negative according to the CAMP test, while the agalactiae is resistant to the bastracine antibiotic test, and its positive CAMP factor. In the second table, we can show the difference between the two alpha-hemolytic streptococci, Streptococcus pneumoniae, and viridin streptococci. Both of them produce the alpha-hemolysis, but Streptococcus pneumoniae is sensitive to the optogene antibiotic list and produces zone of the inhibition equal or larger than 40 millimeter, while the viridin is resistant to the optogene antibiotic disc. Now, they are another differential test used to differentiate group D Streptococci Enterococcus species from other Streptococcus species, which is called bile acetylcholine test. This test is very simple. It uses the bile acetylcholine agar, and if the dark black color it produces, it indicates the positive result. If they are no black color, it indicates the negative result. Why this happens? Because in this test, use the media that are called the bile acetylcholine agar that contain the bile salt, acetylcholine, and ferric acid. The bile salt in a media inhibit the growth of many gram-positive bacteria, allowing only bile-resistant organisms to grow. And if the bacteria are able to hydrolyze the acetylcholine in the media, it breaks down into the acetylcholine, and the acetylcholine reacts with a ferric citrate in the media and produce the dark brown to black color. Therefore, the black color of the media it indicates the positive result. And Enterococcus species such as Enterococcus faecalis are bile acetylcholine positive because they can grow in a bile and hydrolyze the acetylcholine, whereas most other streptococci are negative because they cannot tolerate the bile salt and cannot hydrolyze the acetylcholine in the media. And that's all about this lab. In the next lab, I will explain the antibiotic sensitivity test, the procedure of this test, and the importance of this test in microbiology lab.